Hello, gentlemen. Welcome to section 7.7 called Denaturalization. How do proteins in foods change? Now, the first thing we'll talk about is what proteins are. We might have heard of common examples such as keratin, which is what our hair is made of, collagen, which are what our ligaments are made of, antibodies, which are weapons of our immune system, and hormones such as insulin or many other hormones that we've heard of, which regulate a lot of different processes in our body, and enzymes, which regulate our digestive system or many biological processes in our bodies as well. These are examples of proteins. So what a protein actually is by definition is down here. A protein is an organic substance made of a long chain of amino acids. Now let's look at what an amino acid is. An amino acid is an organic molecule that has both the carboxylic acid and amine functional groups. Now let's talk about what a functional group is. A functional group is simply an atom or group of atoms that give a molecule its characteristic properties. So something that makes that atom stand out and give it, specific properties and characteristics. For amino acids, it has two functional groups. One, which we've explained, is the amine group, which is the NH2 here. And the other one is a carboxylic acid group, which is the carbon double bonded to one oxygen, single bonded to another oxygen, which is attached to a hydrogen. So this is your carboxylic acid group here, or carboxyl group sometimes called, and your amino group or amine group on the left side there. Now there's also a hydrogen above this carbon, and also attached to the central carbon here is this R group. Now R is just a placeholder for right now. R can be many different things. We call them side chains. So there's always something hanging off of this amino acid there. There are 20 different amino acids because there are 20 different. And here are your different R groups, which we call side chains. They're here in green. So each amino acid, as you see, has the same base structure up here, but there's always a different side chain at the bottom. I'm not going to have you memorize all these side chains. You will in a biochemistry class or a different chemistry class in the future. But for right now, just know that there are 20 different ones that can an amino acid can have. These side chains all here. Now, if we look further into the structure of an amino acid and further into the structure of how proteins are created, we have to think about how these amino acids work together. So different amino acids can combine together to form long chains of amino acids, which we call polypeptides. Now, this forms through a process called dehydration synthesis. Now, in this process, we have two amino acids. I have one on this side and one here on this side, the blue and the green one. They have very generic R groups. I'm not sure what they are. Let's just say they are nothing at this point in time. These two amino acids come together to form a long chain through a process called dehydration. Dehydration is the absence of water. So we're going to remove water, and we know that synthesis, sorry, it's spelled wrong, but synthesis is to put two things together. So we put two things together by dehydration, removal of water. So we have an OH group here. This OH group is going to attach with this hydrogen over here. That's OH, and an H, that's H2O. So out comes H2O when that OH and H here that I've highlighted and circled break off. And then this carbon here attaches to this nitrogen here. And then they come together through a covalent bond. We call that a peptide bond. Now, as we form more and more peptide bonds between our different amino acids, we get a long chain of amino acids like this. We have these peptide bonds being formed here, 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 and so forth, making a very long chain. And these polypeptides are known as proteins. Now, these polypeptides, or these proteins, can be twisted, folded, and turned in all different directions, resembling kind of a balled-up shoestring or a balled-up, whatever the case may be, something that's really complex and balled up. There are different structures that can come of this, from these peptide bonds being formed. At first, at the most basic level, we have here our primary structure. This is our primary structure. It's simply a long chain of amino acids. Now, then, this is primary. This never changes. But proteins can also form, and mostly will form, these other types or other structures. The first one we'll talk about is a secondary structure. This is kind of an alpha helix structure, we'll call it. It's kind of just this primary structure here, just coiled in on itself a little bit in this S-like fashion. But think of it in a more three-dimensional space. The other type of secondary structure is this one here. It's a beta-pleated sheet. We have two separate peptides, and they're attached together through hydrogen bonds. We've talked about those before. So different connections here across the way that connect these two peptides together. Now, hydrogen bonds are very important in keeping the this primary structure intact and turning it into these other different structures. Our third structure is a tertiary structure. Tertiary structure is just when the secondary structure further complicates itself and has more hydrogen bonds and more different forces and other different forces holding it together. And the quaternary structure, the last one, which is not as common, is when you have more than one protein, these two lighter ones and the dark one in the back, all coming together. So more than one protein coming together through different hydrogen bonding or other intermolecular forces holding them together. Structure is very important because structure determines function, and the function of proteins can change. So environmental changes like temperature or pH may affect the shape of a protein by affecting the bonding that is responsible for the protein shape. We said the bonding responsible for the protein shape are those hydrogen bonds, and for example, in our beta-pleated sheet that we saw last slide. Now, these environmental changes force the protein to adopt a new shape. So they force the protein to adopt a new shape. This process is called denaturation or denaturalization, as your book calls it. This can be defined as the process during which a change in a protein shape occurs as it unravels and expands outward, still keeping its primary structure but changing its function. So it's important to know that primary structure stays the same, but in the denaturation process, the other structures, the tertiary, quaternary, and secondary structures, can change. But the primary structure, that long chain of amino acids, remains unchanged. Now, denaturation happens oftentimes when a protein gains heat energy. The attractive forces, aka those hydrogen bonds, start to break, and that protein starts to unravel and form a long chain of amino acids. Now, denaturation, a little bit more about it, it's not a change in composition, meaning your primary structure, how those amino acids are connected, stays the same, but the structure changes. And since structure determines function, denatured proteins behave quite differently from their original or undenatured forms, which are commonly called active protein form. Proteins are denatured at about 40 degrees Celsius, and at higher temperatures they may even begin to break up or bond with each other and form clumps or aggregates. So at higher temperatures, sometimes even that primary structure can get broken up and broken down. Cause complications, and sometimes not complications. We'll talk about that now. So how does denaturation connect to our lives? Well, the first examples we'll use are with food. When proteins are heated and denatured, they are extended, and are more vulnerable to attack by enzymes, which are catalysts that aid in digestion of your dinner. Enzymes attack at a specific point called the active site, and here's a picture here to display that. So, for example, when you're cooking meat, when we have raw meat, it's all balled up and it's not a denatured protein. It's in its active protein form. When we cook it, however, we straighten out the protein, and the enzymes in our stomach can attack that meat a little better and break it down a little bit better for digestion. And the same is true with eggs. When we have a raw egg, it's in its undenatured state, it's all balled up, and when we cook it, it denatures, it lengthens, and that changes its function. We use raw eggs and cooked eggs for different things. We don't use them for the same thing. And lastly, another example that's not with food is Alzheimer's disease. This is a disease that plagues many of the elderly in our lives, or people that we know of. And what happens here is that in the brain we have proteins that, due to certain conditions in the brain, they will slightly denature. When they slightly denature, that means that chain, that protein, that balled up protein structure starts to unravel just a little bit. And when it unravels a little bit, there are different chemicals in the brain, different things that can come in and attack that site that is now exposed. When it's exposed, it oftentimes cleaves or splices. It gets cut, so that protein gets cut in half. And then it happens again and again and again. And these protein pieces that are no longer big, long chains, but now little aggregates, we call them, or amyloid, they congregate or aggregate in a certain place in the brain. And with that, they start to kill different brain tissues, different neurons, different synapses, and that leads to symptoms that go along with Alzheimer's disease. That's a little bit about proteins, gentlemen. Please take notes. Hasta mañana.